Welcome to Virology. I'm Vincent Racaniello, and I am going to be your professor for this entire virology course. And this is lecture number 1, What is a virus? A little bit about me before we continue. I have been working on viruses since 1976. I've been at Columbia. I'm at the medical school campus. I've been there since 1982. I have a lab that does research on viruses. I've been teaching about viruses. I've been teaching this course now. This is the 11th year. And I've written a textbook and done a lot of other things. It's my goal to teach you virology, to teach as many people in the world as possible all about viruses. We live and prosper in a cloud of viruses. Viruses infect every living thing on the planet. There is no life form that does not have some kind of a virus that infects it. In fact, most have many, many different viruses. Humans regularly eat and breathe billions of virus particles a day. You're breathing them right now. They're floating around in the air. You eat them with your food, as we'll see. And we carry viral genomes as part of our genetic material. Every life form on Earth has viral genomes as part of their genetic material. It is just remarkable. Virus discovery, which was once driven only by disease, all of those early viruses that I showed you, yellow fever, poliovirus, smallpox, they were all driven by saying, oh, there's a new disease. Let's find out what's causing it. And they found viruses. Nowadays, we just look for viruses by genome sequencing. And here's a fascinating study published a couple of years ago, where investigators collected a variety of insects, crustaceans, nematodes, mollusks, small critters, I would say, and they extracted their nucleic acid and sequenced them, and they found 1,445 new viruses from 220 invertebrate species. And this is what we do now. We simply extract nucleic acid and we look for viruses and we find them by the score. We find thousands. Just doing a study like this, you can find thousands of new viruses. And this has hardly been done for any species. Like rodents, we hardly know what's in rodents. And they're a threat to humans because any animal that lives in large numbers and close to humans is a threat for a pandemic spillover. You know, you think about all these viruses and someone's asking, how likely are they going to be able to cause severe disease? Who knows? You want to take the chance and have a pandemic like we have now? Obviously, most of them don't go anywhere. But now and then, the right one is going to get into people. It's going to spread a little bit in people and then get better adapted. We'll talk about that in a moment. If a single hunter is bitten or somehow gets infected and doesn't see anyone for 6 months, that's the end. That's the end of the infection, whether or not the hunter gets sick. If someone is harvesting bat guano in a cave and gets infected and never sees anyone else, that's it. However, if that farmer harvesting guano goes home to his family and infects the family, now the virus may start to pass among humans. Maybe it gets selection for better human passage, and then maybe if one of the kids in the family goes into town, then the kid will spread it to more people. You get the picture. The bottom line here is that we are nowhere near ready enough for the next pandemic, and there's going to be one. There's going to be a flu pandemic. There is going to be another coronavirus pandemic, probably in 10 years or so. And I hope this experience makes us ready. I'm not sure. Caves can have millions of bats in them. And if you sample, the easiest way to sample bats is to go in the cave. You should be gowned up to protect yourself. You can collect the guano, the bat poop that's on the floor of the cave, and bring it back and sequence it. And you can find thousands and thousands of viruses, but it's barely been done anywhere. And you know, one thing that's quite interesting is that many farmers in parts of the world collect bat guano to fertilize their fields. So you can immediately see how they can infect themselves with some viruses. If you take human feces and you sequence the nucleic acid, most of the RNA virus sequences, 91% of the RNA in human feces is similar to plant viruses. So we eat a lot of plants. They have their own viruses. We have them in our feces. They just pass through. They don't infect us. You know, you can have your fecal virome done now. You can send feces to a company and they'll sequence the virome. Sounds like a Harry Potter curse, right? You can look in dirt. Many companies take dirt and make a fermentation in the lab and see if there's anything in that. My wife used to work at Merck, and whenever we went on vacation, she used to collect dirt from various places. She would get a GPS coordinate, put it in a Ziploc, and bring it back. And if they ever found anything, they could go back and get more. The coronavirus was called coronavirus because when they were first looked at in the EM, someone said it looks like the solar corona, this fringe of spikes around the envelope, and so they called them coronaviruses. And when that first virus was isolated from patients in China, early 2020, they did an EM. They wanted to know, what do we have here? They had done a bronchoalveolar lavage. They took a little of that fluid, put it under an EM, and boom, looks like a coronavirus. Do you know, last year, one of the neighborhoods in New York City with the highest infection in the country was Corona, Queens? You can't make this stuff up, you know. So someone wants to know about the movie. It's a graphically produced. It's all CGI, right? And they, consult virologists to know what the intermediates are, and then they do the animation. It's like how you make a movie now, and except it's with a virus. And there's a really cool animator, Janet Iwasa at the University of Utah. She does this for a living. She does animations of proteins and viruses. It's very cool. Right? It's kind of sci-fi-like. I love it. For years, we couldn't grow viruses in cells in culture because we didn't know how to do cell culture in the laboratory. That didn't come out until 40s and 50s, actually. We'll talk a little bit about that today, but we had to use animals of various sorts, like chickens and ferrets and rabbits and non-human primates, etc., rodents, dogs, cats, whatever people could get their virus to grow in. And they would make stocks of viruses in animals. For example, polio virus, for many years after its discovery in 1908, people made stocks of the virus by inoculating non-human primates. As soon as they got paralyzed, they'd remove the brain and spinal cord, grind it up, and they froze it, and that was their stocks. Actually, they didn't freeze it because there were no freezers. So what would they do? They would inoculate another animal. And they kept doing that to maintain the stocks of viruses. Can you imagine? And here we have, in one case, we give 100,000 PFU intraperitoneally. Right, we put a needle in the belly of the mouse. These mice are fine. Every one of them survives. I put 100,000 PFU intraperitoneally last week of a human enterovirus into mice. They all lived because the virus doesn't infect mice. But in this case, if you put 1 PFU intracranially, that means right in the brain. It's very easy to do in mice. The skull is soft. You can put a needle right through it, especially when they're younger. 1 PFU is enough to kill the mice. So apparently, the virus cannot get from the peritoneum into the CNS. But if you put the virus right in the CNS, only 1 PFU is enough. So virulence depends on the route of inoculation. And there are many, many variables that control virulence. So it's pretty cool stuff. And you can eventually imagine that we're going to have a whole catalog of susceptibility genes. We're going to sequence your genome at birth. And it's a good idea. It's going to be stored somewhere. And your physician can look at it and say, oh, you're at risk for this. We have to do this for you and that. And this is going to be part of medical care in some point in the future. We're not supposed to live beyond reproductive age, right? We're not supposed to live past our 30s because that's what we lived to before medicine. And medicine prolongs our age. And as a consequence, we have cancers and Alzheimer's and susceptibility to infectious diseases. So everything's a trade-off in life, right? You want to live longer? This is what you have to deal with. So people will figure this out eventually. Now, how do you diagnose influenza? Most of the time you go to your provider and they look for influenza-like illness, which means fever, cough, or a sore throat. And if it's here in New York in the winter, they say you have flu and I'm going to give you a prescription. Now, of course, this year it's different because there's not much flu around. But in most normal years, this is what they would do. They could give you a rapid antigen test, but they're not very good. And it's better off for them to do it symptomatically and give you a prescription, because we do have some antivirals. You can do PCR if you'd like. It's going to take a little bit longer, but and nobody does viral culture anymore. But you can do serology, of course, but that's going to be later on. Now, it's not easy to figure out how many people have had influenza. So the CDC makes estimates, because you don't diagnose everyone. You never do. You never diagnose everyone for any viral infection. And so here are the estimates for the 2019-2020. That's last year. Because this year there's like nada, zip. It's amazing. In this graph, we're looking at pneumonia and influenza mortality. So you can't often tell if people have died from influenza, because they don't write on the death certificate influenza. They may write pneumonia. Sometimes they'll write pneumonia secondary to influenza, and that's great, but they don't always do that. So you have to make estimates. So here are estimates of, it's called P&I, pneumonia and influenza mortality. And here on the lower right, I put in this year's map for this week. Oh, we're in March, right? Same month. Nada. Look at that. Minimal activity throughout the U.S. This is the site where you can find all this stuff. Really good stuff. And we suspect that masking and distancing and all that stuff has really knocked down influenza activity. I'll be interested to see the final numbers on predictions of death and so forth. It's not easy to tease out, because COVID causes pneumonia deaths, so it's going to be hard to separate those two. That's not very scientific. But anyway, these are some of the things people say as reasons for not getting vaccinated. Kids should get infected naturally. Really? With the possibility that they die or have debilitation? It's ridiculous. I can't afford to immunize my kids. Well, that may be an issue. It should be free. In fact, in many countries it is free. It should be free in the U.S. I don't see why it's not. In some cases, you can have a medical exemption to vaccination, but they should not exceed 1% of the population. In some places in Texas, the medical exemptions are 50%, because you go to your doc, I don't want my kid vaccinated, they give you a medical exemption, and they shouldn't be. And if they don't, they lose their patients. It's horrible. There are also religious exemptions, which should not be allowed at all, in my view. Many states are outlawing them. It's just another excuse not to be vaccinated. The rabbi says, don't take the vaccine. You should be naturally infected. What does a rabbi know about vaccines and infectious diseases? They listen to their leader and they get infected. Here, Hare Krishna community in North Carolina, a church in Texas was critical of measles vaccination. I would like to talk to the leader of that church and say, what do you know about vaccines and epidemiology and disease? So he tells his congregation to not get vaccinated, and they get sick. It's totally crazy. This drives me nuts. Anyway, there are outbreaks all over the country like this, and that's why we have measles throughout the years in the U.S. still. There was a big outbreak in Westchester last year. Same thing, Orthodox Jewish community. The rabbi said, don't get vaccinated. Those of you going to be doctors, you're going to have to deal with this. You better tell people to get vaccinated. Don't give them a medical exemption unless they need it, like if they're immunosuppressed and so forth. The COVID vaccines are not licensed. They are authorized. Pfizer, Moderna, and J&J. Pfizer, Moderna, mRNA, of course, J&J is an Ad26 vector. They are authorized. They have emergency use authorization because the phase 3 usually takes 2 years to complete it. It's been accelerated because we're in an emergency. And some people have used that as an excuse to say they're not safe, but that's not true. All the safety concerns really are addressed already. What's addressed in 2 years is the durability, not safety. It's the durability. So we don't know how long they're going to last because we haven't gone 2 years. So if you don't take your drugs, the virus will overwhelm you. You got to keep taking them. So this is not a big deal to change the vaccine every year. People are freaking out with COVID vaccines. Oh my gosh, we're going to have to make a new vaccine every few years. Just read the literature, folks, and learn what's been done. And then you have ongoing studies in people where you observe for many years to make sure there are no untoward effects. And in the case, always safety is the overriding concern when we do this. only 1.4 million kids get polio, which is an unfortunate side effect. And in fact, in the U.S., this is the cases of polio in the U.S. from 60 to the present. In 1961, we introduced oral polio vaccine, and that eradicated wild polio. Last year, if a wild case of polio in the U.S. was 79. That's the straight line. But it replaced wild polio, with vaccine-associated polio. All these gray bars are cases of kids who got paralyzed from the vaccine, up to 20 a year. And so in 2000, that was no longer considered acceptable. So we switched to inactivated polio vaccine. And now no more polio caused by the vaccine. I don't think this was ever really acceptable. It's a real debate in the field. And sometimes you can have a computer just churn through the active site and model compounds to fit in, and then you pick the best hits and you synthesize them and see if they actually work. And as I said, this is all done by robots. On the left, these lines are each a virus genome in a tube. I give you a tube of virus, and there are lots of particles in it, and that's what the genomes look like. Everyone is different. They have different kinds of mutations scattered throughout it. Now, on the right is the sequence of poliovirus, which I did back in the 1980s, and I got that. That's one sequence, right? It's not right. That's not what was in the tubes that I was sequencing. That's a consensus, because the sequencing techniques I used give you an average of the population. So you have to really work very hard to get the quasi-species to find out how every RNA or DNA is different. Really hard to do, because it turns out that sequencing has an error frequency associated with it. So it's very hard to distinguish that from the quasi-species. Nevertheless, many other studies have shown that this is what a genome looks like, not this. This is what I call the myth of consensus genome sequences. For any given RNA virus population, the sequences cluster around a consensus or an average, but every genome can be different from the other. And when you do a sequencing technique, you typically get a consensus or an average, unless you do high throughput deep sequencing, and you may be able to get individual molecules. But as I said, it's hard. And in fact, this consensus that I published for poliovirus, which many people do for other viruses, may not exist in the population. It's just an average. That's the myth. This is important for understanding what happens. The more of these you do, I'm going to be a little cynical here. The more of these you do, the higher profile journal you can get your work published in. All right. Now, one of the first descriptions of an emerging virus was in the 60s. This book called Fever by John Fuller. This described the emergence of Lassa virus in Nigeria. And this is a book that got me to be a virologist. I know I've told you that before, but I just was looking for something to do as a recently graduated biology major. I was working in a lab. I read this book, and that was it. You have this book to thank me for teaching you today, so you might want to read it. It's really good. A lot of it takes place at Columbia. Here's some of the activities that promote virus emergence. A lot of people, of course, but also, I mean, look at this travel. This is I took this screenshot at some website where you could see all the planes in the air at any one time. This was done in 2019. And obviously, air travel brings viruses everywhere, but also other things you might not think of. You know, dams, water impoundments, irrigation, deforestation, rerouting of wildlife migration, wildlife parks, long-distance transport of livestock and birds. Amazing how much viruses they carry. Of course, blood transfusions, xenotransplantation, putting animal parts into humans, societal changes with regard to drug abuse and sex, huge increase in drug abuse and so forth. Urbanization, daycare centers is a big one. Hot tubs not properly sanitized, air conditioning because of the water used for chilling, and then, of course, the used tires, which I've told you about, transport mosquitoes. And don't forget climate change. So let me end with this slide, which is a summary of the estimated burden of chronic viral infection in humans. So here we have the number on the y-axis in millions. And endogenous retrovirus, that's everybody on the planet. Every human has an ERV, multiple ERVs. So this is everyone. And then you can see nearly everyone has all of these viruses, anelloviruses, HHV 6, 7, VZV, EBV, CMV, polyoma. Almost everybody has polyoma, JC and BK, adeno-associated virus, herpes simplex, and then it starts to go down, Hep B, papilloma, Hep C, HIV is way down here. All right, why am I showing you this? Well, first of all, a lot of people have persistent infections. And we each harbor at least 8 to 12 of these, but more because we don't, all the viruses that are in us, we don't even understand how they're reproducing. And more on top of it, our virome is on top of it. These are the 8 to 12 herpes and polyomas that we understand. But what it means is if you're studying a disease, you never have a control population that is uninfected. And so if you're just studying, say, diabetes or some cancer or some neurological disease, you can never separate the role of these viruses in that disease. You have to think about it. You have to understand the potential contribution of these viruses to whatever disease you're studying, but you will never have an uninfected control population. So you will never know if the virus that are in these people is impacting the disease or not. And I just want to leave you with that. Think about that, because you'll see lots of studies in the literature. This gene is causing diabetes. You just don't know what the viruses are doing. I want to thank you for taking this course. It's been my pleasure and honor to teach you. And the only thing I ask of you is not to forget, 90% of knowledge is knowing where to find it. You know where to look it up. And you're familiar now with virology. You actually know more virology than 99.9% of the world's population. And so don't forget it. Again, be curious. Always want to know how things work. I do a lot of communication, and I always exhort people to be curious. And by the way, if you're interested, I do a bunch of podcasts besides virology, microbiology, parasitism, immunology, and there's our lectures that I post as well. Neuroscience. I do a blog. I'm all over social media. So follow me somewhere and see what I do. Don't forget to be curious and be passionate about what you do. And HIV causes AIDS. There is no question about it. I get emails every day. People who tell me HIV does not cause AIDS. It's never been proven. The virus has never been isolated. Blah, blah, blah. It's a bloody waste of time. Get with your life, folks. These are called AIDS denialists.